Hi there. Good evening, everybody. Noswaith dda. I hope you can see and hear us okay. This is Anna Holmes and I, and we're ready, I hope, for the first in a series of webinars from Amgueddfa Cymru about species in the Nat Tran Byth project. We are going to start with the PowerPoint presentations now, which should explain everything as we go along. So I will just get our excellent technical assistant Josh to put me back onto PowerPoint, and hopefully you can see the first slide there. Okay, we've got quite a lot to get through, so let's get going. As I said, Anna Holmes is seated here, and she is our ACE curator of marine molluscs, and I'm Ben Rosen, I'm the curator of non-marine molluscs here at Amgueddfa Cymru. So we're both mollusc specialists, so that's the one thing we know really well. In this webinar, we will be going until about 8 o'clock. We've got a lot to get through, but there will be a gap in the middle, there'll be a break, and there's plenty of chances for you to go and step away from the screen. We won't know if you've done that because we can't actually see you or hear you, but it should all work fine. So first, there's a bit of a housekeeping bit. Then I'm going to explain why we're doing these webinars and how they came about. Then I'll go through the 3 snails that feature in the Nat Tran Byth project and hopefully introduce you to each of those. Then we have a break after any questions. Then we're going to come on to the bivalves, so that's the clams essentially. We've got a native oyster and an Arctic pea clam to talk about, 2 very different organisms. And then finally we'll have a few more questions and discussion at the end and see how it went. So here's the housekeeping part, that's what they call it, isn't it, at these meetings. First of all, please mute your microphone if you haven't already, so we don't have too much sound interference. Secondly, feel free to ask us any questions as we go along. If you could do that using the Q&A function or the chat function. Sorry, I'm just peering at the screen. I think it's either called Q&A or chat. We'll try and answer them as we become aware of them as we go, or we'll deal with them in the middle or at the end. If you would like to have simultaneous translation into Welsh, please could use the translate function on Vimeo, which should be available on your screen. And finally, this webinar is being recorded and we'll put an edited version of it online sometime in the next few months so that other people can look at it too. So why are we doing these webinars? We're delivering them from Amgueddfa Cymru, from the museum, for the Nat Tran Byth project over the next few years. And we're using the National Museum's collections and our expertise on natural history to, first of all, show people how to recognise these species as though this were a training course, like the kind of training courses we run up and down the country about identifying particular groups of animals, plants and fungi. Secondly, to show people where and how these species can be found. Often knowing exactly where to find them is the key to success. So that'll be a bit like a field trip to some very hard to reach parts of Wales in many cases. And thirdly, to show people what the project Nat Tran Byth is doing to help them. And one of the great things I think about Nat Tran Byth is that a lot of the species in it are quite obscure. They're not well-known species, they're not famous very often, they're quite difficult to find, and they can be quite difficult to recognise, or both. They can be difficult to find, recognise, or both. Nat Tran Byth, if you haven't heard of it, is a major conservation project funded primarily by the National Lottery Heritage Fund, but also by Welsh Government, the Arts Council, Esme Fairbairn Foundation, and the Landfill Tax Community Scheme. It's run by Natural Resources Wales in partnership with 9 other organisations. The museum is a bit of an extra here. And it's a brilliant project, it's really exciting, it's already in full swing. And it's aiming to improve the conservation fortunes of 67 rare and threatened species in Wales. And they're all kinds of different species, which is wonderful. It's right across biodiversity. It's through a large number of projects, 11 of which are place-based in particular parts of Wales, and 10 of which are kind of nationally coordinated, so they work across Wales for species all over the place. And molluscs, our favourite group, come up in at least 4 of them as target species, named target species. So we've got Swansea Bay Coast Commons and Communities, Welsh Marine Treasures, Penllyn ac Ynys Môn, and Llysau Mynydd Eryri, which I've been saying as carefully as possible, Snowdon's Mountain Jewels, Snowdonia's Mountain Jewels, all have particular molluscs that are part of the conservation goals of those projects. So that's Natra an Byth, the project introduced. I'm under the impression that many of you might not be aware that the museum here in Cardiff and across Wales is actually involved in nature conservation and natural history. So we've prepared a little video, which we're going to cut to now. And that should give you a bit of an introduction to what we have in the museum and the kind of activities we undertake. Welcome to Cardiff, the capital city of Wales. It's great to be in the middle of town and out in the park. And just over there is a building where you can see stunning natural objects from all over the planet and the entire natural history of the world. It's Wales's very own Natural History Museum, and we're going to show you a bit about how it works. This building has been open for over 100 years. Every year we have more than 250,000 visitors. Have you had a look yet? On display we have showcases of Welsh habitats, animals and plants from every part of the globe, including some beautifully crafted wax models, some exquisite rocks and valuable minerals, and we tell the whole story of the evolution of Wales. And yes, we do have dinosaurs. Exhibitions are only a part of the work that we do. The objects you see on display are only the tip of an iceberg. Behind the scenes, we've got over 5 million more, and they're used in all kinds of research projects, events and activities. To find out more, we need to go behind the scenes.
Mae ein curaduron wrth ei bodd yn siarad am y gwaith sy'n digwydd tu ôl y llen yn y amgueddfa. Weithiau mae pobl yn dod i weld y casgliadau fel hyn. Dyma lle rydyn ni'n cadw yr specimenau sydd wedi eu cadw mewn alcohol, fel mwydod, cregyn, crancod, cymwchiaid, sermôr a'r mwy. Mae llawer o specimenau yn dod o ymchwil amgueddfa. Mae anghenfilod o traethiau Cymru gyda ni. Mae pob gwrthrych yn y amgueddfa yn dweud stori.
Insects are the most diverse animal group. For instance, there's over 250 species of bees in the UK, and only one of them actually makes honey. Also, they're very fiddly. Organising a scientific insect collection requires a lot of work and skill. Microscopes are essential, whether you're a student, a trainee, or an experienced entomologist. Thousands of new species of insects are still being discovered. At this museum, we have described hundreds over the years. The first specimens of each one are preserved and remain available to other scientists to study and borrow.
Like all scientists, we publish our results in international journals. The museum has published over 1,000 papers on its collections-based research.
A museum is the best place to compare one species with almost any other, and we have the most complete collection of Welsh flora and fauna in the world. We've used the collections to make several identification guides, which are used well beyond the museum. This, for example, is the go-to guide for British cockles, mussels and clams. We've even used them to produce guides for other countries whose biodiversity is less well known. We can also identify non-native species that have recently arrived in the UK and could become a threat.
Rhai o'n specimenau mwyau poblogaidd yw'r vertebratau, anifeiliaid gydag asgwrn cefn fel adar, mammaliaid ac ymlusgiaid. Mae llawer ohonyn nhw wedi cael eu benthyg gan amgueddfeydd arall, orielau ac ysgolion. Hefyd, mae gwyddonwyr yn defnyddio'r specimenau ar gyfer samplu DNA. Mae hyn yn helpu ni i ddysgu mwy am poblogaethau anifeiliaid ledled y byd. Mae llawer o'n specimenau wedi dod o zooau a pharciau anifeiliaid fel Bili y Morlo a Bryn y Teiger.
In Wales, we have 800 species of moss. We can collect mosses in Wales from sand dunes to mountains, and we dry them and we put them in packets. And with a little bit of water, we can rejuvenate these collections and study them and figure out what they are and tell us about what the environment was like where they were collected from. Our specimens help us look at how distributions of species change over time, and we collect these and we use our databases to map our collections and see these changes.
Diatoms are single-celled algae less than 0.5 millimeters long. The museum has a collection of more than 7,000 samples stored on glass slides. We are studying diatoms to monitor pollution and climate change in rivers and lakes.
This is the fossil of a little dinosaur called Dracoraptor. It was found by some amateur collectors and we were lucky enough to have it donated to us. Little Dracoraptor was one of the first carnivorous dinosaurs from Wales. The bones themselves are fairly well preserved and they have been three-dimensionally scanned so that the public can see those on our website and rotate them and look in each direction.
That's a quick look around, but this building is only the beginning. Our work takes us far and wide, from fieldwork in remote regions to installing exhibitions at other museums and a wide range of public events around Wales. We look forward to seeing you soon at Wales's Natural History Museum, and get in touch if you have any questions. Hwyl am y dro! Hello again, I hope you enjoyed our video and it all sounded okay. So back to the molluscs. So the first group of them are going to be the gastropods. That's probably a term you've heard before if you're familiar with nature and biology. The gastropods are the snails and slugs. In the waters in the sea around the UK, we're about 4,000 species, a very large number. But there are a few that only live on land and a few that only live in fresh water. And the ones we're going to be talking about in Natra and Byth are actually all terrestrial ones that live on land. The relevant features of this group, the vertigo, the whorl snails, and many others in fact, are that they're very sensitive to moisture levels. They can only live in places which have a particular humidity in the soil, in the air. They tend to be detritivores, so that is animals about whose diet we know rather little, but they eat mainly dead things. They can live in tiny habitat areas and they can survive there for many generations, less than 10 metres squared. They can also breed very quickly and they have a special trick up their sleeve, which is being able to reproduce without having a partner, without mating. And a lady called Eva Sharland, who did a PhD here at the museum on these species, actually found that the Welsh populations in many cases predominantly reproduce without mating. One problem that snails have is they're slow moving, which makes them very poor at reaching new sites or even to recolonise old ones. So birds or butterflies or even some plants might quickly recolonise an area from which they've been lost, but that option isn't always open to snails. After those, we're going to come on to the bivalves, clams, mussels, cockles and so on, and the oyster and the Arctic pea clam are great examples of that. There are a lot of species in the sea and a few in fresh water, but these guys can't live on land, of course. In the case of oysters, they're filter feeders, and in fact almost all bivalves are filter feeders, so they draw water through their bodies and extract any nutrients from it. The oysters can't move, they attach themselves to a hard surface and that's it for life then. However, for a short phase of their life, when they're larvae, they're spread potentially quite long distances, hundreds of kilometres sometimes, in the plankton which floats at the surface of the sea. They live in the sea. The pea clams, which live on land, in fresh water, separated from other bodies of fresh water by large areas of land, are also filter feeders, so they strain the water through their bodies. They're often able to move to a limited extent, so they have a little foot that comes out like a little toe or a tail, and that allows them to climb around to a limited extent, so they can move from one part of a pond or lake to another. But their larvae don't travel such vast differences. In the case of the pea clams, the larvae are kept quite close to the centre of the body. They're brooded, as they say, and then they're just released over a short distance. So they too, rather like the snails, find it tricky to colonise new places sometimes. And they can be very sensitive to pollution in the water and other environmental changes such as the water temperature. So that's the end of the introduction. I'm now going to cover in a bit more detail the vertigo whorl snails. So I think I warned you it was going to get quite detailed quite quickly. Now we'll see just how much. So what we've tried to do is introduce all these species to a wide audience all in one go, while still telling you enough information so that you get that those who already know the species will learn something new, and also you'll find out what's going on right this moment with the Natra and Byth project. So the vertigo species, the whorl snails, they have an English name, whorl snails, but I'll probably hardly ever use it because I call them vertigo. A lot of people call them vertigo, it's just a different way of pronouncing it, and it really doesn't matter which one you say. Vertigo means whirling in Latin. So first of all, how do you recognise them and what are their lookalikes like? Well, in Wales, sorry, let me just tidy that up. We actually have 10 species in Wales out of the 11 that are in the UK. There's a sculpture of one there on the top right, which is, I think it's probably supposed to be a vertigo species. It's in the city park next to the museum here in Cardiff. And it's reasonably accurate. Shouldn't have the bottom two tentacles because those have been lost in vertigo species, but never mind. And it's in a reasonably appropriate habitat because it's in an open space, a grassland, and that's where you find vertigo, in meadows, dunes or wetlands, especially when they're relatively unspoilt and undisturbed. They're quite rare in woodlands, most types of fields, arable or pastures, in moorlands, and in urban habitats. They're very rare. The big thing about vertigo, or the small thing about vertigo, is they're all about 2 millimeters long, which makes them smaller than sesame seeds. So I've got a picture of sesame seeds there, if you're struggling to remember just how small they are. But those sesame seeds are bigger than most of the snails I'm going to be talking about. And that makes them difficult to notice, even when they're very numerous in the environment. The only one you're likely to bump into by accident, and indeed the most common in Wales by quite a long way, is the common whorl snail, Vertigo pygmaea. It's nice when something's got common in the name and it actually is common, and that's the case here. So that's the one you're most likely to see. So very few people in Wales will have seen most of these species, which I'm going to show you now. And to try and give you a sense of what it's like to be here at the museum or on one of our training courses, I've set up a microscope with a camera on it, and I've recorded what you would see through the microscope and how it would be to manipulate these tiny specimens. So there it is set up, and then on the right that's an ordinary brown garden snail, which I'm sure you recognise. The kind of tools you need to manipulate these tiny things could be pointy forceps or stalk spill forceps, are very useful things for this kind of stuff. Even a paintbrush to push them around, and these little gelatine capsules, like the kind of thing they put medicine in, are very useful. And that's how we maintain the collections here at the museum. I've included a label there because, as we all know, the most important thing is to label your specimens so you don't get mixed up as to when and where they were collected. So, all right, another type of video here. Let's hope this works okay. I've got a video of what you would see through that microscope looking at the shell of that brown garden snail. So hopefully this will play. I might have to press space. So there's the shell, and taking it away, you can see the rest of the sample from which that shell came, and that's all underneath it. So you get a sense of how much smaller they are than the brown garden snail. On the right there, we've got a nice sand dune snail, a wrinkled snail. Just push that out of the way. This one in the middle is a door snail, the kind of thing you find on trees, cliffs. Move that one out of the way as well. And you can see we've got some glass snails there, the vitrinids, but the vertigos are the smallest of all, and there they are, less than 2 millimeters long, so smaller than those sesame seeds. And you can see a few features of it under a decent microscope like this. There are also some baby vertigos in there, which you can also survey and count too. So that's what they look like. At the museum, we have done a number of training courses on these species, and we have slides which have all the species set out, and a couple more besides. Now, if I were to go through this entire thing, then we would need a 4-hour webinar or something, and I don't think anyone wants that. So I'm just going to introduce you to the ones that you're most likely to find with them, and that are most often mistaken for the target species. Here I'm going to talk about Loria, Cylindricea, Pupilla muscorum, and show you how much bigger they actually are than the vertigo, for which they're sometimes mistaken. So I've got a mixed sample here. Here comes my paintbrush, and I'm just going to push the Lorias over to the left. So you can see they're about just over 3 millimeters long, but they're much, much bigger than the vertigo, and a rather different shape as well. And there, that one I'm pushing at the moment, is a baby Loria. So this comes about whenever you're studying snails, you will find juveniles of the same species. You find juvenile shells. And the way to match them with the correct adults is to realize that the juvenile shell will eventually grow into the adult. So it will fit, if you take my meaning, it will fit into the top part of the adult shell. So we've got 2 adults and 1 juvenile Loria on the left. Then in the middle we've got an adult pupilla and 2 juvenile pupilla, and then on the right the vertigo, the adults and the juveniles. And you can see the juveniles are a very different shape to the adults that they finally grow into, but they are nonetheless characteristic. So you can match them up by shape there. So very often when people are sending in records of snails, or they're identifying them for the first time, they find a loria and they think, oh, that's small, it must be a vertigo, but often it isn't. So there's a summary of those differences with the shells all arranged alongside one another. You'll notice there are also differences in what we call the teeth, which are in the hole, the mouth or the aperture of the shell, and those are like little projections of the shell that obstruct the aperture to a different extent. Loria's just got a single tooth, Pupilla's got a single tooth, although it's not always present, whereas vertigo on the right there tends to have a lot more teeth, multiple teeth. If you get into snails, and I heartily recommend that you do, the Field Studies Council book on how to identify them is excellent, and there's a really useful laminated guide that the Conchological Society has produced as well, and that's a nice kind of aide-memoire to all the species that you're likely to see in Britain or Ireland. So here we go. Let's go through the 3 target species one by one. First of all, the narrow-mouthed whorl snail, Vertigo angustio, as we like to call it. And this was first found in Wales by John Gwyn Jeffreys of Swansea. There he is, his magnificent smouldering presence with those awesome Victorian sideburns. He was one of Wales's best ever conchologists, malacologists, people who study mollusks. And he discovered this species on his own back doorstep, if you like, in Swansea in 1830. However, it has always been a rare species. And thankfully for us, it's a relatively easy one to recognise. So here, I've got a mixed sample of lots and lots of specimens of Vertigo pygmaeus, and then in amongst them are 3, there's one there, 3 specimens of the rarer Vertigo angustio, and I'm just pushing that to the right. So there we go, there are 2 of them. Where's the 3rd one gone? It's a baby, there we go. I'm pushing it up to the top. So there are 3 of those, and they look rather subtly different to the others, I expect you'll think. But once you've done this a couple of times, you'll realise this is actually one of the easiest species of vertigo to recognise, because it always coils the other way. So on the left there we've got the Vertigo pygmaea, which is very common, and that always coils to the right hand side, which means that when you've got the pointy bit upwards, the aperture or the mouth is on the right hand side at the bottom. If you look at the Vertigo angustio, you put the pointy bit at the top and the mouth is on the left at the bottom. And once you get your eye with this, it's like left and right handed screws, you can't mix the 2 up. And in almost all circumstances, a left-handed vertigo that you find in Wales is going to be a Vertigo angustio. It's also, helpfully for us, a bit more rugby ball shaped, so it tends to be narrow at the top and narrow at the bottom as well, which is an extra feature you can use. So this species, it was described from Swansea by John Gwyn Jeffreys in 1830. As far as we know, it's extinct in that area. It has been searched for, but there's very little suitable habitat left, of course, because Swansea's there. It also lives in other places on the Gower Peninsula, area of outstanding natural beauty, and in Carmarthenshire, where there are some magnificent sand dunes. Part of Natran Byth with the Swansea Bay project and the support of BugLife and NRW has been to do some surveys of some of these areas to find out what the numbers of snails are like in these areas. And just earlier this year, Martin Willing and I went off to Pendine and we did a survey of the Ministry of Defence land there. So the results of that will be out in due course. So how do you find them? You need to go to the right habitat. This is a classic habitat, I would say, for Vertigo angustio. In the distance, you can see a salt marsh in front of the woods. And then in the foreground, that salt marsh is gradually transitioning to a kind of sand dune environment. Lovely places like this on the Gower. And this is how you look for them. You have to get there and you have to get close to the ground. You can do it in all conditions, and of course, once your fieldwork's been arranged, that's what you have to do, no matter what the weather's like. So this is Vertigo expert Martin Willing out there in pouring rain last summer while we were looking for them. And you can find them just using your eyes if you've got very good eyesight, but very often you need to use some tools or some kind of assistance. So you can use sieves, pair of wooden screen sieves there through which you can rub a bit of vegetation, and the snails will fall through sieves of a certain size. And here we have somebody from NRW sieving them again on that very, very wet day on which we covered a lot of sites. And we did find a few specimens in the field by doing it this way. Their microhabitat, think about how small these snails are. Their world is very small, and so it's a microhabitat which is particularly critical to them. This is a video of what it might be like if you were a snail and you could see the microhabitat. It's a very dense thatch of all these short, wiry strands of festuca grass. There's other plants in there. There's lots of detritus at different stages of decay, which is what they'll feed on. And it's a very humid environment. Even on a dry and sunny day, a lot of moisture is trapped close to the sand and soil surface by this type of vegetation. There's a shortcut, of course, and I love shortcuts. I use a shortcut to find these snails, and I've been testing this method out with Martin to see how effective it is. And that's using a suction sampler, which is just essentially a vacuum cleaner. And I'm just showing you how it works there. You just turn it on and then prod it into the vegetation. That's Karen Wilkinson from NRW having a go there. And that is a quick way of getting a sample like this. And this is actually the sample that I showed you under the microscope in the first instance, which contains all kinds of life: spiders, beetles, thrips, springtails, everything. It's unharmed, it's sucked up, and you can put it onto a tray and you can have a look to see whether you've got any snails in there. And I can see a vertigo right in the middle. Brilliant. That's one of the better samples. You can see it in the field using the suction sampler if you get lucky. Just to increase my chances of actually spotting things, I tend to resort to these loop things these days, which you can get in all kinds of different shops, but they're useful for all sorts of different things, but they provide a bit of extra magnification, even for people who already wear glasses, so that they can get a better look in the field. So that kind of work for this species has been, we've done a lot of it already. Martin Willing has been leading on that for BugLife and NRW. He did a big survey in Whitford Burrows in the North Gower in 2021 and looked at a large number of sites, and the red spots there where it was found. So it was only found in a few parts of the reserve, but the range of habitats that it was found in was actually rather wider than we expected, and they included some places with a bit of willow or Corsican pine in, which came as a real surprise. And if you're interested in this subject, these places or this species, Martin has got a full-length YouTube talk on this subject, which he gave to the Conchological Society last year. I would recommend watching that to get a bit more information. So this species, what's actually threatened by? Well, those habitats are fairly well protected, but they do change. Sand dunes are always changing, and there's always a risk that they're going to be outshaded by trees and scrub, and the bare sand and the more open areas are going to be replaced by trees and scrub. And remember, these vertigos don't like woodland very much. So it's quite important to maintain some open habitat, particularly in the dune slacks, so it's the wetter parts of the sand dunes, where these snails get the humidity they really need. There's a rainbow there, which came out at the end of that really wet day, which was great to see, and that kind of exemplifies the way the results have been going so far. We found lots of new subsites for this species at Whitford, and Martin even found a whole new site for it at Nicholaston Burrows on the Gower. It's also excellent to find out that the habitat range for this species is a bit wider than we once thought. It probably means the populations are larger and a bit more robust, but there's still more to come on that. At the bottom there, I've mentioned that there are Natural Resources Wales reports about each of these surveys and the species account by Mike Howe, which give criteria on how its conservation is measured. So quite a lot is being done to keep an eye on this species. Right, number 2, Vertigo mullinsiana. Do we have any questions yet? No, I'm not seeing any questions. Good, I shall carry on. Vertigo mullinsiana, or Desmoulins whirl snail, as very few people call it. This is a voluptuous snail. I've called it that because of its shape, and it lives in luxuriant fens, so a very different habitat from Angustior. It was only found in Wales relatively recently, 1998, by a member of the Conchological Society called Barry Colville, who was a really keen and skilled amateur snail fan. And here they are under the microscope, along with some of those common vertigo pygmaeas. So again, there are 3 of the rare species amongst several of the common ones. And I hope you can see that the rare species in this case is a lot larger than vertigo pygmaea. So it's voluptuous, it's curvier, it's shinier, it's kind of like an overweight version. It also has a flaring lip around the edge of the mouth, which is quite a distinctive feature. And there it is on the right hand side there, against the common vertigo pygmaea, and you can see it's a lot larger, it's more inflated, it's the fattest vertigo you're going to find. It's got a glossy shell and that flared lip, which actually connects the teeth in the mouth in a distinctive way. So actually this is another unmistakable vertigo species. One thing you might get it mixed up with is the one in the middle there, which is called vertigo antivertigo, strange name. That's another species that you might find in similar habitats, and actually in Wales it's more widespread than Willensiana, but it's a lot smaller and it has different teeth, so usually quite easy to distinguish the two. So Vertigo willensiana is actually quite an important species in recent British conservation history. If anyone's old enough to remember the Newbury bypass protests of the 1990s, where people formed a camp in the trees to try and stop a road being built, and this guy Swampy became quite famous in the media then, and stayed for many weeks, I think, resisting the developers. One of the reasons they didn't want the road to be built was because it would threaten populations of this snail, and indeed they had to do a lot of mitigation work around Newbury in Berkshire to create new habitat that would support these endangered populations. And if you look at the map on the left there, you can see that this species really most common in lowland in England. There aren't many populations of it in Wales at all, but there are one or two. That's the kind of classic habitat there on the left. There's a really rich, luxuriantly vegetated marsh next to a lowland river, in this case the River Thames, where we went to for a Conchological Society meeting this summer. Surveying for it is rather straightforward. You can do it all in the field. You don't need to collect any samples and sieve them or anything like that. You can just look closely at the vegetation, or especially at this time of year when the snails are climbing up the stems, beat or shake the reeds over a white tray placed on the ground, and you can simply count the snails in the field and then put them back where they've come from. And if you do a count like that, you can estimate the number that you get per square metre or half a square metre, judging by the number of trays. So actually surveying it and surveying its numbers is relatively efficient. In Wales, the best site by far is in the Llŷn Peninsula, not far from Pwllheli, where you've got a lightly grazed fen meadow with rushes growing in it. Again, Martin Willing's done most of this survey work, and he's found out which fields it's in and which fields it's probably not in. And that's at Cwsgech near Pwllheli. It was also found nearby in the Afon Penros, which is where they've done that more detailed survey work, and they found that it's actually there in huge numbers. It's one of the largest and most robust populations that we have in the UK of this species. So despite not being in lowland England, it seems to be doing rather well. But like the Angustia I was talking about before, it is threatened by changes to the habitat. So again, there's the threat of scrub encroachment, so trees growing in the place and gradually turning it into a woodland. Also, the density of the vegetation that it is living in is important too. So you need to have some grazing by animals, just the right level and the right kind, to maintain its population. Thirdly, it's under threat of the fields being drained or dried out, so you need to maintain the hydrology. And this was a persistent factor in conserving those populations in lowland England in the past as well. And again, there's lots of reports about that. So finally, the third whorl snail I'm going to talk about is Guyer's whorl snail, Vertigo guyeri. I couldn't come up with a nickname for this easily, but I have called it a vanishing relic of post-glacial landscapes, because this is a species that we know was a lot more common in Britain when it was very, very cold and just after the ice had retreated at the end of the last ice age. This was only detected in Wales in 1988 by somebody called Boyce, and then it was re-identified by Barod Colville. It's another species that's always been rare here. And this one is a lot more difficult to identify, and it's also difficult to be certain that you've got it correctly identified in the end as well. So again, we've got 3 specimens there amongst the common Vertigo pygmaea. One of the distinctive things about Gaiarai is it's got fewer teeth, and the teeth are very weak, but that's difficult to make a judgment on alone, because the pygmaea can vary quite a lot in how strong its teeth are. So you need to collect specimens and do a proper comparison. Here they are side by side. Another thing about Vertigo pygmaea is it usually has quite a strong crest on the side of the shell. I'm not sure if I've got a pointer. No, I haven't got a pointer, but if you look at the side of the shell, you can see there's a kind of white stripe down the last whorl of the shell there, and that's quite a strong crest or rib, which you wouldn't see in Vertigo Gaiarai. I hope you can see that the pygmaea has got 5 teeth, I can see there, whereas the Gaiarai's only got 4, and they're not connected to one another by another part of the shell. They stand alone because they're quite small. Also, the Gaiarai shells tend to be very relatively dark, and they're more barrel-shaped than the pygmaea are as well. They tend to be a shorter and broader shell. But they really are very rare, so you're unlikely to find them by accident. There's another species, Vertigo liljeborgii, which is also very rare, and that lives in similar habitats in Wales, and it's absolutely critical that the two are properly distinguished, because they're both rare, and we would like to know of any other populations of either. So the best thing to do if you're really stuck on the identification is to take some photos or send the specimens to a specialist such as myself or Martin to get them checked against other authenticated examples. So this is their habitat. Now, not nearly as luxurious as what we saw for Melinsiana, or as picturesque, I don't think, as what we saw for Angustior. That is a kind of a particular type of soggy pasture, which has got open seepages where the water is actually seeping out of the ground. So they need to be very, very wet and very, very bare, just like the country would have been when those ice sheets retreated about 8,000 years ago. So this is quite a rare habitat, and it's easily destroyed. It can be destroyed quite rapidly by sedges simply growing in it and then shading it out, and then it's no longer suitable for Vertigo Gaiarai. And this place is Cwrs Gerch on the Llyn, and the species was last seen there in 2008. Martin and Mike Howe have been there to do a lot of survey work in recent years, but they haven't successfully found it there, I'm afraid. So this is the map of all the known sites in Wales, and that Cwrs Gerch is at the bottom on Pen Llyn. The other 2 sites are on Ynys Môn, Anglesey. It wasn't found again in recent surveys in Cwrs y Greiniog, but it has been found in Waun Eirad, if I've said that correctly. So it's still alive at Waun Eirad. So it's down to 1 site in Wales, and it really is on the edge. In recognition of that, this species has been used to define the special area of conservation in the Anglesey Fens. It's a primary feature for their designation. They've also got marsh fritillary and southern damselfly, which are beautiful and rare insects, and they're much better known than this snail, but the snail is actually much rarer than either of them. That's the habitat of Waun Eirad, the last remaining site. Again, very short, very soggy, very unmanaged or unimproved for agricultural purposes. It's a tiny site, it's less than 200 metres wide, very vulnerable to drying out and any changes in management. But they have confirmed it's still present there, and it's the last population in Wales. And the way you could look for it is demonstrated by Martin here. He's cutting a little vegetation, and then you can run that through a sieve or take it back to study carefully to see if there are any specimens present. So that's threatened by a lack of grazing, which means the open seepages get shaded out by growth of plants. It's also threatened by overgrazing. If the livestock take all the vegetation, then there's nothing for the snails to actually eat. And also, again, the hydrology. So if the sites become either too wet or too dry, then the species will disappear. All this data comes from the NRW evidence reports, which are available online from the NRW website. So that was a whistle-stop tour through the 3 vertigo species in Nat Llanbith. Angustio, which is doing rather well in the southern dunes, which is great to see. Mulinsiana, which seems to be holding fast on Pentleen, although we don't have any new sites for it yet. And Vertigo gyri, which is still living on Anglesey, but is right on the edge. So that is the one that personally I'm most concerned about, because I think that's one of all the molluscs that I know of in Wales. This is one of the top 3, I think, that is a threat of disappearing in our lifetime. So whatever Nat Llanbith can do to maintain it will be most welcome. So I hope you learned something from that. Does anybody have any questions about it? Oh, it looks like I've got a question. That's not a question, but thanks anyway, John. No questions, but loving the content, John Clark here. If that really is you, John, then thank you. It's time for our break. If you do have any questions, then ask them after the break, and I'm sure we'll be able to do our best to answer them. We'll see you in about 10 minutes. Go and have a cup of tea or whatever else it is you drink at this time of night, and we'll see you back with some oysters. Thanks a lot.
Hi everyone, we're back and we're going to be talking about oysters for the next 10 to 15 minutes now. In particular, Ostrea edulis, which is our native oyster. I'm going to talk about how to tell it apart from the non-natives. But first of all, what is a bivalve? Because Ben's been talking about tiny, tiny snails. Bivalves are mollusks, and instead of having one shell, they have a shell of 2 parts, or valves. And these are joined together by an elastic ligament. In the images you can see on the screen, if you look at the top 2 images, you can see a small brown band, and that's the elastic ligament, and it kind of acts like a hinge or a spine on a book to let the valves open and close. Bivalves also have a foot, so some are rather mobile, like cockles, and they can move around using their big fleshy foot. But in other species, like mussels, the foot is greatly reduced. They have siphons. Ben just talked about that about half an hour ago when he was talking about mollusks in general. And these siphons, there is an inhalant and exhalant siphon, and they also have teeth. So on the hinge line, so again, the 2 shells at the top of the screen, these are the insides of the valves. Right at the very top are various kinds of little shelly protrusions, and those are called teeth. And those are how we actually distinguish various families and superfamilies. And bivalves also have 1 or 2 adductor muscles, which allows the shells to open and close. And those adductor muscles leave scars on the inside of the shell, which are diagnostic features as well. And there are around, about 11% of the mollusks are bivalves. But there is a great variety of them, but we're going to talk about oysters. So oysters have been eaten for thousands of years. When the Romans invaded 2,000 years ago, they brought with them a hunger for seafood, and they took to our common oyster. They even shipped UK oysters to Rome. But when the Romans left, their enthusiasm for eating oysters left with them. But by the 1400s, they were back in favour as a staple cooked in ale and pepper, or pickled for travelling inland. In 1864, over 700 million oysters were consumed in the UK, which is quite remarkable. And the oyster industry employed many, many people, especially in Oystermouth on the Mumbles near Swansea. So the oystermen would go out in their little fishing boats and dredge for oysters in the daytime. And after dredging, they would return to the shore and tip their catch into what was called an oyster perch. You can see the 2 images on the screen. The top one is one taken in the late 1800s, and the arrows are pointing out the oyster perches. And they were basically divisions measuring about 16 square foot, and they were formed by stones. And each area was marked by a floating log. So the oysterman would leave his catch there to feed while they were required for sale, or either sold locally or transported on the railway for sale elsewhere. Some of these perches are still visible today along the beach near the inshore lifeboat station. In the mid-19th century, dredging of oysters was popular until overfishing exhausted the beds and pollution nearly drove the populations to extinction. Artificial breeding has saved the species here, and there are many restoration projects around the UK. So this is the species in question. This is the European flat oyster, and you can tell it apart. You can see the irregular subcircular outline. The top valve on the left is flat, and the outside of it has irregular flattened comarginal ridges on it. The bottom valve on the right hand side is gently cupped, and on the inside you can see these tiny little crinkles that are called comata, and they actually travel all the way around the inside margin of the shell, and that's quite a diagnostic feature for this species. They have one adductor muscle scar, which is right in the middle, and that is kidney shaped. And this is the outside of the bottom valve as well, and you can see these irregular radial ribs on it. But how many oyster species are there in the UK? Well, we only have one native oyster, but there are unfortunately 2 non-natives. And there are also 2 deep water species and 3 rafting non-natives from the Caribbean, but we're not going to talk about the last 2 groups. So in each of these sets of photos, the Pacific oyster is on the right hand side and our native oyster is on the left hand side. This is trying to compare the 2. The Pacific oyster has a more elongated oval outline and a really solid shell, which has strong zigzags at the edge of the margins, and both valves are cupped. Remember, our native oyster has one flat valve and one slightly cupped valve. And on the inside of the Pacific oyster, there's no sign of the little comata, the little crinkles that Ostrea edulis has. Ostrea gigas, the Pacific oyster, can reach sizes of 18 centimetres in length. The Pacific oyster was introduced via Canada in the 1960s, and it grows twice as fast as our native. Because of the 2 cupped valves, there is more meat inside, so it's much more commercially viable. But it is farmed in great numbers in the UK, and it has established in the wild as well. It is taking over habitat that our native oyster could potentially inhabit. In Wales, it's spread around Pembrokeshire, North Wales and Anglesey. So this is the second non-native species. This is the Chilean oyster, and it's a lot more similar in shape to our native. It has one cupped valve and one either flat or very, very gently cupped valve. The outer part of the shell is dark brown to black, and it sometimes has tan rays on it, and occasionally it has random projections of the shell, which you can see on the image on the right hand side. It reaches a maximum size of around 8 centimetres, compared with our native species, which reaches a maximum size of 10. The inside of the shell is a creamy to yellowish as opposed to white, and it was introduced in the Menai Strait in the 1960s to be farmed, as it too grows a lot quicker than our native oyster, but populations didn't take off, and it was given up as a species that could be farmed here. However, it is expanding slowly in its range in North Wales. Its dispersal is limited by being a direct developer, so that means that the female broods the larvae inside, but once released they have a very, very short pelagic phase, and this is very much unlike Ostrea edulis, our native oyster, which has highly mobile larvae, and it allows for a greater settlement distance. Again, I'm going to show you some young specimens of all 3 species as well, just to compare. So even when they're around 2 to 3 centimetres, you can still tell them apart. The image at the top shows Ostrea edulis and Ostrea chilensis, the Chilean oyster. The Chilean one you can see is still quite dark at that size, and you can see the rays on it as well. And the Chilean one is a lot more ovate in outline, where ours is more circular. Gigas, the Pacific oyster at the bottom, again is still elongated, and it's almost teardrop shaped when it's small, and the shell is a light cream, sometimes with a reddish mottling. So what are the threats to Ostrea edulis? Well, in the 1800s, overfishing reduced numbers, and then pollution almost finished it off completely. And it has been difficult for the oyster to make a comeback. Oysters are filter feeders, and so any pollutants will be inhaled and have a negative effect on the animal. Larvae and juveniles are killed off by the Ostreid herpes virus, but the main disease affecting Ostrea edulis is bonamia, which is an internal parasite that causes mass mortality, and it's spread by moving infected oysters around. It can live in Pacific oysters and mussels without killing them, but they just spread the parasite to healthy native oysters. Ocean acidification inhibits shell growth, and it also disrupts the digestive system of oysters. And invasive species take over habitats and bring with them new diseases, which our native species are more susceptible to. And climate change brings a whole host of problems, influencing susceptibility of oysters to fluctuations in water temperature and nutrient concentrations. And this, teamed with pollution, is affecting aquatic organisms more than scientists first realise, and it accelerates food web simplification as well. Excuse me. So why are oysters important? Why should we try and help save them? Well, they stabilise sediments and they provide stable habitats and unique ecosystems and create mini reef habitats for other animals and plants to live on. They can also filter water really well. So one individual oyster can filter 200 litres of water a day. They're easily farmed and they're a sustainable food source. And as a bonus, they're carbon sinks, locking calcium carbonate into their shells. Native oyster restoration projects are underway around the UK, and even the Navy is getting involved as well in the Solent. But the Pacific oyster represents 98% of those harvested for sale in the UK still. Many oyster farms in the UK farm Pacific oysters because they grow much quicker and they have more meat. But there are some native restoration projects spread around the UK and around Europe, and Ostrea edulis is even being farmed in South Africa, the Northeast Pacific and the West Atlantic. So there is good news for the species. Sadly, we can't do a lot about global climate change, but you can make some changes to your lifestyle to offset things like your carbon footprint. Also, food choices we make have an impact. Cheap and fast is not always the ethical way to go. So if you eat oysters, buy local and buy native. Other things you can do to help out is to help monitor oysters on our shores, both non-native and native species. If we can prevent further spread of gygas, then that gives edulis some hope. So if you are interested in recording oysters, I'm going to show you a little field trip video now and help you how to know how to spot oysters in the wild. So we're going to go to Mumbles, and I'll just see if I can get the video to work for you. All good. So we're going on a virtual field trip to the Mumbles now to look for oysters. As you can see, the pier and the big wheel are useful landmarks here. So on the shore to the left of the pier, you can see the remnants of the old oyster perches used by oyster fishers. That's me pointing them out. And Ben's just about to zoom in on them. The old oyster grounds are just below the watermark there. And then to the right of the pier is the rocky pebbly shore where we're more likely to find live oysters today. Just offshore there's an oyster farm that grows native oysters, and it's likely that these are seeding this shore as well. And we're here in mid-March during a spring tide to ensure that we can see the oysters above the low water mark. And here we are. Can you see it? One live oyster. There you go. one gently cupped valve and one flat valve. This large one is an empty one, but you can see the cupped valve has gentle radial ridges on it. And there's the top of the flat valve. This is an oyster faker. It's a saddle oyster. And this is a slipper limpet, which is an invasive marine snail. But when small, it could be mistaken for juvenile oysters. And here's some more real oysters. You can see the radial ridges quite clearly there. And there's a large one on this outflow pipe as well. You can see the flat upper valve and the curved lower. It's nearly time to head back now, but this caught my eye. Underneath the pier, there were mussels covering the posts of the pier. But no oysters attached, it's too high out of the water. But below this were lots of empty mussel shells and empty oyster shells as well. So what's going on? And then we spotted the culprits. Starfish, looking rather fat and well fed. They love eating bivalves. Here's one just finishing its meal. Their tube feet grip onto prey, and then their stomach is thrust into their prey to liquidize their meal. The stomach is then pulled back into their body. There's an oyster survivor. Hopefully there are a lot more out there. And that's the end of our trip. It was a quick one, but hopefully it's given you a bit of a taste for looking for oysters.
Brilliant. Thank you, Anna. Excellent stuff. So one last species to cover, and then we're done. Slideshow. Beginning. Okay. Right. Okay, I hope you can see that. A very different bivalve indeed. This one doesn't live anywhere near the sea. In fact, it couldn't get much further from the sea in Wales. The Arctic alpine pea clam, or Conventus conventus, is, I would suggest, one of the most obscure of all the species that are featured in Nat Rambith, and that's why I particularly like it. It's part of a project run in North Wales called Slysau Mynydd Eryri, the mountain jewels of Snowdonia. And I suppose if it's going to be a jewel, then this one would be a pearl because it's got that link to the oysters. But it probably doesn't really look very special. It was first found in Wales in 1915 by this guy Charles Oldham, who is a Manchester-based, I think, naturalist, and he was very, very good at spotting tiny differences that other people hadn't noticed. Which is essential when it comes to pea clams. They really are a problem, and if you've ever done any freshwater biology or pond dipping, tried to identify what you find, you may have come across pea clams, and you've probably been told by another naturalist that they're almost impossible to identify. And I have to say, I probably agree with them for the most part. We've got about 30 species of freshwater bivalves in Britain. The swan mussels are big enough to hold a pint. They're about 30 centimetres long, but the smallest of them are these pea clams. There are about 16 species, and they're often neglected by biologists, even those who already like molluscs. And there's a few reasons why they're particularly difficult to identify and study. I'm not trying to put anybody off, I'm just warning you that they're not the easiest of molluscs to get to grips with. First of all, they're tiny size, less than 5 millimetres, and a rounded shape, which makes them quite difficult to handle and examine. They're always rolling around, like the snails were earlier. They have few distinctive or obvious features on the outside or on the inside, and they vary as well, like all molluscs do, in their appearance, which doesn't help. Sometimes you need to open them up to see the hinge or the pores on the inside, and these features, they're there, but they're very difficult to photograph, which means communication between people who are interested in them enthusiasts is actually hampered a bit. It's quite difficult. But last of all, and this is, in my experience, this has been one of the biggest problems, is about 2/3 of the British species are very widespread and very tolerant of different conditions. So it's very difficult to rule any of them out. They all look very similar, they're all very widespread, and they're all very tolerant. So how do you know which ones you have? There are too many options. So massive respect to Nat Tran Byth for daring to include one of these species as one of their target species. And I'm really glad they have done, because it puts the Arctic pea clam on the map for perhaps the first time. There it is, a nice picture of one. It's an Arctic alpine species, as the name suggests, so it lives only in cold, clean lakes and streams, either in high mountains or the very far north. And I'm sure you're familiar with the other kind of species that are in Tlysil Mynydd Eryri, which are things like the mountain saxifrages and the Snowdon lily and things like that are only found at high altitude. It's got a tiny, very simple white shell, only 3 millimeters long, so just a bit bigger than the vertigos. And very recently indeed, it's been classified in a genus of its own by some colleagues in Russia who published the results there, and you can't see the details of it, but that's one of those family trees based on the DNA, and they found out that it's actually quite distantly related to all the other species that we get in Britain, which I've annotated on that diagram. And so it's been given its own genus, Conventus conventus. So it's actually changed name since Nat Tran Byth has started. We're learning all the time. So if you do some fishing and you come up with some bivalves, small bivalves that is, the biggest of them you'll see are these orb mussels, this brown one here, and they're about the size of a fingernail or something. In fact, they're sometimes called fingernail clams. They're quite easy to see. These are some smaller ones with a kind of little cap on the end of the shell, if you can see what I mean by that. And that's called the unbone of the shell, that little area. But that's another distinctive one. That's what's now called Spherium lacustri, which is a lake orb mussel. And those two species are fairly chunky, fairly distinctive, so not too difficult to deal with. You wouldn't find them in the same habitat as Conventus, though. This is to give you a sense of the diversity, the remarkable diversity you get of these Pisids, and how difficult that is to get to grips with. So I'm going to give you a kind of conveyor belt experience from a single sample here in the museum, where over half of the UK fauna was found in a single sample, at a single spot in the River Thames. And this is a particularly valuable object to find in the museum. I felt like it was the Rosetta Stone or something, because it consists of a single sample which has been sorted into the species and kept together, so you can see exactly what makes one specimen different from one other. I may not be explaining that very well, but in practical terms that's a really useful thing to have, because having the specimens labelled individually doesn't really help that much. You need to see them together to see what makes them different. So I'm going to try and share that with you by showing this video. So we've got Pisidium amnicum at the left, that's the biggest one, that's the easiest one. And there we go, 9 different species, and there are different numbers of each one. And as you can see, superficially, they're all really similar, but they've all been carefully sorted into these different groups. And the numbers of each vary, so they vary in their abundance. So there's this whole community there living in the riverbed of these tiny clams that almost everybody is too afraid or unable to identify. And yet, there they are. So it was Ian Killeen that sorted that sample, and he's one of the co-authors on the standard freshwater bivalve book, and it's really useful to have specimens like that preserved in this way. Okay, so they're tricky. I might add that there are large numbers of these individuals, and it's a bit of a paradox, but the more specimens you have, the easier it is to sort them out. If you imagine you've got a drawer full of mixed cutlery, it's easier to sort them into different groups once you know that some of the features crop up again and again than it would be if you just had one example of each one. I'm looking at Josh here to see if my metaphor makes any sense. Okay, so that's the River Thames, loads of species in there. If you go to a high upland lake up in Eryri, up in the mountains, you are not going to find that many species, if you find any at all. Harsh environments, not ideal for molluscs for all kinds of reasons. But here we have 2 species that Anna and I found in a particular lake. One of them is Conventus conventus, and the other is Eugleza liljeborgi. The larger ones are liljeborgi, and they're quite. They have a rougher surface and they're quite grooved. So the liljeborgi there are on the left, and the conventus are on the right. Now, I myself am very uncertain sometimes about the identification of these things, so I've checked those with Martin Willing, who's one of the experts, and he agrees that they're liljeborgi and conventus, which had been recorded from that lake before as well. In case you think we're imagining this, you can look at different specimens from different collections from these localities and others, and you find that all the conventus look the same. And in fact, you can go all the way to Novaya Zemlya, I think it's called, which is in the high Arctic of Russia, very, very remote place. And the shells look the same there. And it's those shells from Russia that were the basis of that polar biology study, which gave this species its own genus. So when I finally get round to it, I would love to DNA barcode the specimens we have from Wales and check that they're identical or very similar to the ones from Novaya Zemlya. I suspect they are, and if they are, then that's confirmation that we're dealing with a single species. Two of the other things you might confuse with it are the red-crested pea clam, and God, look at that map there. This shows that this is one of those really widespread and tolerant species that you can find almost anywhere. It can even live in puddles that dry out during the summer, and superficially it looks almost the same as Conventus conventus. However, it's never been recorded in any of the lakes that have conventus in Wales. And I've looked right back into the detailed notes of Charles Oldham and the others to be sure that that's the case. On the inside, that has a couple of features. There's a feature in each valve which is called a callus, and is diagnostic if it's there, but I have to admit it's very difficult to see, and it's difficult to photograph as well. Really quite difficult. So, Conventus, where has it been found? Well, there are 4 known sites in Wales. The last record before us was in 2000, so you could say that our records are the first ones this century. It hasn't been seen in England, where it was found in the Lake District, since 1938, so it's perhaps already made extinct there, but it probably hasn't been searched for properly recently. So we compiled all the Welsh records we could find, and they included a lot of unpublished ones that we have here at the museum from Peter Dance, who did some of this early collecting, some from the Conchological Society, and also from Mike Howe at NRW, who's been maintaining them. So there's only the 4 known sites. There's 2 on Yr Wyddfa, the most southerly block of those mountains, 2 on the Glyderau, the middle one, but there were no sites on the Carnedd Llewellyn in the north. And all these lake habitats are remote, very remote, usually well over a mile from the nearest road, and fragile, and they've got multiple levels of protection as well. This is the one above Cwm Idwal. So Cwm Idwal, you may well have visited there. It's a fabulous natural amphitheatre with these crags around it and a lake in it. Well, if you go just above the main part of Cwm Idwal, you come to Cwm Clyd, which is an amphitheatre above an amphitheatre, and that has a tiny little lake in it. And that's where Anna and I went in 2022. There's the lake. And the bottom of this lake, and this is really important, this seems to be the classic, the critical thing for this species. This kind of habitat, which the botanist Robbie Blackwell-Miles calls a lawn community, I think that's right. And that's a really interesting idea, which has all kinds of cultural associations as well in North Wales. But this is like an underwater lawn, and it really does look like that. Through these crystal clear waters, you can see these short plants growing in amongst the stones. And then in the summer, they come out and flower. The stones and the kind of nature of the stones and how granular they are seems to be important to the clams, because they can only live between rocks of a certain size. And of course, the chemistry of all these upland lakes in Snowdonia is also really important. Some are heavily polluted by copper and other metals, whether that's natural or due to mineral extraction, so they'd be unsuitable for molluscs. Others seem to be okay, and we're just not really sure why they don't have much in them. But it was present in Cwm Idwal, in Llyn Clyd, which is above Cwm Idwal. And there's Anna. Anna's daughter came with us to take some of these photos, and that was great. And we found loads and loads of Conventus there. It was actually abundant in that pond. The first time it had been recorded for a long time, but we found it relatively easily. And we took some specimens, but did little harm to the population. But the lake itself, of course, is a tiny habitat and thus very fragile. The next record came from Robbie Blackall-Miles himself when he was visiting Llyn Bach in Cwm Glas on Yr Wyddfa. It's important to give these names in full because there's another Cwm Glas and there's certainly another Llyn Glas, and there are lots of Llyns and Cwms with similar sounding names or the same name, these clams are in some of them and not all of them. That is Llyn Bach in Cwm Glas, where Robbie found it to be very common, and that's a photograph he took there in the top right. So he was able to find them himself, and he's given us some specimens so we can check the DNA of those as well. These lakes aren't, as the crow flies, they're not that far from one another, but if you need to hike to them, it's quite a serious undertaking. So, for someone like me, where I want to start at the bottom rather than spend the night on the ridge, and you can only visit one every day or so. Robbie and I had a great time in June this year. He took me up to Llyn Glas in Cwm Clogwyn, not Llyn Bach in Cwm Glas, but Llyn Glas in Cwm Clogwyn, and Llyn Coch. Two very interesting lakes, very close to one another, on the western side of Yr Wyddfa, Snowdon. You can see the railway from there. There were snails present in those lakes, but we didn't find any bivalves, and we did have a really good look. This is a known site for Conventus, so it's a bit concerning that we didn't see any sign of it. Not even any empty shells, which you can often find in places where a mollusk has recently disappeared. You still find the shells for some time. And we did find snails there. Finally, I wanted to go to somewhere on Carnedd Llewellyn, which has got, in fact, it's got the highest lake in Wales. This isn't it, there's an even higher one. It's got some very high lakes that may or may not have been properly investigated for this bivalve before, and could potentially play host to it if we looked in the right way. So Robbie and I went up to Llyn Ffynnon Caseg in Cwm Caseg, which is on the Carnedd Llewellyn. And that was a long walk to get up there. I would say it's the most remote place I've ever been to in Wales, and it was absolutely fantastic. Well worth walking up there just as a hiker. But we did not find any mollusks at all in that lake. So I suppose I could say I can't say I wasn't warned because there were no mollusk records from that lake, but when there are no records, how do you know for sure that people haven't really looked? Well, this is a good example of a negative report, but I still think that's quite useful because it could have lived in that lake and have been overlooked. So we have now checked 3 of the 4 known sites while Natran Byth has been going on. And we found that the species is still common at 2 other sites, which is excellent. It's doing okay. It has not been found at Llyn Glas and Cwm Clogwyn. That's a bit worrying. It may have disappeared from there. If it has disappeared, we would like to find out why. I do wonder if this is perhaps because this is the lowest altitude site, only 530 metres. So possibly it's the most vulnerable to rising temperatures, because Robbie tells me there are members of the Arctic alpine plant community where the lake temperatures has a critical value, and if it goes above that, then it will not survive anymore. And one might expect that to be true of this Arctic alpine species. So we're not sure about that. But we didn't find any on Carnedd Llewellyn, and it hasn't been found in any of the larger lakes on the eastern flanks, which are mainly reservoirs. So we don't really know why that is. It might be the chemistry. There's one other known site to check, which is where that John Bratton record came from. That's near Glydyfach, but again, it's quite a remote and difficult to access place. So we found out as much as we can at this point. I think we're looking for it. There's a lot more that could be done. There are loads of lakes in Snowdonia. I would love to know what would happen if you could sequence these species and then use eDNA methods to find out how much more widely they would occur. But now I've changed my perspective on it. Now I've seen the species in the field and gone to some of these sites. I do think it probably is just about as rare as we think it is, and it's not likely to be hidden under a lot of hiding in places where it could easily be found. So, what do we think it's most threatened by? Well, it's definitely threatened by a warming climate. It's a bit like Vertigo gyri that I spoke about in the first half. It's one of these glacial relics. It's going to be the first set of species to disappear as the climate warms. So it's certainly threatened by a warming climate. And Robbie's got all kinds of really good ideas about keeping an eye on this. Nutrient enrichment. I saw this for myself. You go to any of these lakes and there are Canada goose poos all over the place. I don't know whether they're bringing these nutrients up the hill and then pooing them out on the lake shores, or whether they're just feeding on the environment there. But there's certainly an argument to be made for controlling this non-native species in these fragile environments. And finally, there's pollution by visitors, including the wearing of sunscreen. If you go swimming in these mountain lakes, then great, good for you, but try not to fill them with sunscreen. There are alternatives to the mineral-based ones you can get, and awareness could be raised of that. So I've had a lot of these ideas are Robbie's, and I've had some really interesting discussions with him about this species, and it's really interesting that they've included it in that Trambyth, because they're thinking outside the box, I think. If you like pea clams, you will need at least one of these books, and I would suggest possibly both. The Field Studies Guide is self-explanatory. The one about the mollusks of Poland is really useful because it's in English and it's got a lot more detail on some aspects of the Pizids, and I found them very difficult to identify without having at least 2 identification sources at hand and a collection as well. So if that doesn't convince you that they're a difficult group, I'm not sure what would. So what have we covered? We've covered whorl snails, oysters and Arctic pea clams, and we're nearly at the end. I must acknowledge all the help that we've had. I want to say diolch yn fawr to Nat Trambyth and everybody involved in that, particularly John Clark, Cass Crocker, Mike Howe and Karen Wilkinson, all from NRW. Vic Leporevan, Rodri Viney, Guy Fryer and Josh Smith for helping at Amgueddfa Cymru, and our other colleagues who will be producing the next webinar in the series. Robbie Blackwell-Miles from Plant Life, Martin William from the Conchological Society, Claire Dinham from BudLife, and Matt Sutton for doing the botany on the most recent survey we did. And of course all the landowners and site managers who've given access to their land for us to be able to do this field work, and the rest of the stuff that's making Nat Trambyth such an interesting project. They include the National Trust, the National Park Authority and Kinetic as well. So finally, a quick recap on the 5 species, and I'm going to ask again whether you have any questions, and I hope Anna will come back and help me answer them. Hello. Do we have any questions there? Which order do we answer these in now?
Newest one's at the top, so I'd say go from here up.
Alright, so lovely field trip, Anna.
So going back to Vertigo gyri, how easy would it be to rear this?
That I honestly couldn't say how easy it would be to rear Vertigo gyri in captivity.
And Claire Sampson would love to invite you to Ynys Môn to discuss.
Well, I love going there. Yeah, I know that Eva Sharland did a study on them here at the museum, and I think it was Wynarad that she was looking at them from, and she found that about, I think it was 80% of them were aphallic. So they didn't have a male function, essentially. They were just females that self-fertilise themselves. So you'd think that would make them relatively easy to breed, but whether it's easy to find the right food for them or not, I don't know. Whether you'd have to keep them refrigerated, don't know. I'm not that kind of biologist, really, but I'm sure you could find someone who is. Great. And
there's a comment from Drogana. Peeing in the Llyn's also an issue for the pea clam.
Wild swimmers.
Wild swimmers.
Yeah, I suppose I'm a fair weather wild swimmer myself, but I try not to pee in places like those mountain tarns. In the River Ely, I don't think anyone would notice. But, yeah, I think that is an issue, and I think dogs as well, because I'm not quite sure what the rules are on dogs in places like Cwm Clogwen, which are also sheep grazing territory. But yeah, people's dogs just leap into lakes at a moment's notice, and they could be not only weeing in it, but dogs are treated with insecticides and anti-wormers and things like that. So I think in small bodies of water, those things are likely to be big problems.
Would there be potential for the museum to set us some survey targets for Nature an Bith to find volunteers to focus on?
Yeah, possibly. Yeah, I think so. It's difficult because sometimes things are so rare, obscure and fragile, it's hard to think of how they could become volunteer projects. But there are other occasions where, the mixture is just right. So, like, native oysters are quite, as long as you know how to recognise them, they're fairly easy to find, aren't they?
Yeah, they are, absolutely.
You can watch them grow as well, I suppose, can't you? Because they're quite big and they live for years.
Yeah, just go back to the same spot as long as you can remember where you saw it.
Yeah.
Use what 3 words, that's handy.
Oh, right,
Yeah.
Right, yeah. Yeah, we do a lot with volunteers, don't we?
We do.
Anna's got a student at the moment, and yeah, good idea.
Oh, there's another question. Which species is most likely to become extinct? Oh gosh, that's sad.
I guess it would be the vertigo gyri in Wales would be the most likely to be extinct because it's down to a single site, which makes it very vulnerable to some damage to that site or a very bad climatic year. But then again, the Arctic pea clams are vulnerable too. Of course, they've all been here for a very long time, but there's a lot of stresses on the environment now.
Yeah, how much longer can they hold out?
Yeah. Maybe we should be culturing them. That's a good idea.
Not in wine, Irad, please. Surveying the last site could wipe them out.
Yeah, well, I see what you are getting at. I think that if you were to look too vigorously for them, then there might not be any left. That is a risk, but I hope I've shown you how difficult gyri is to actually positively identify. I quite like the suction sampler because it lets you check things in the field, but there are probably even less lower impact ways of finding it as well.
Nothing new's come in
in
the last minute or two.
No questions on the chat either.
No.
Okay, that's great. You can always contact us about molluscan matters, of course. That's what we're here for.
Yeah.
We get all sorts of interesting inquiries, and it's great to be involved in like an active conservation project in Wales, which has still got so many different directions that it could go in. So feel free to get in touch, and you can always come and visit the museum as well.
Definitely.
Yeah, and extra appreciation to Anna, who's managed to shake off a cold deliberately to.
I've come in and spread my germs.
Yeah, that's okay. It was worth it. So, thank you all very much for coming, and I think we can sign off there. Right, we'll see you for the next one, which will be in March. The next webinar will be in March, and it will probably be about insects. So, no more mollusks. Thank you.